Hey everyone, welcome back to the channel. Let us discuss on CNS pharmacology. The CNS is an extremely complex and heterogeneous structure. It is the major coordinating system of the body and source of adjustment of an organism to its environment. It functions as a rapid coordinating system and is responsible for moment-to-moment control of various body activities. It regulates rapidly changing visceral events, muscle contractions, secretions of some endocrine glands, and functioning of body systems. It is more sensitive to effects of drugs than any other system of the body. Neuron nerve cell is the major functioning unit. Other cell is glial cell. Division of CNS and their functions. Spinal cord. It receives peripheral sensory information from skin, muscles, tendons, joints, visceral organs, etc., and conveys it to the brain. It conveys the motor command from the brain to peripheral skeletal muscle. It also performs many functions like regulation of walking movements, pain withdrawal reflexes, and some other reflexes that control blood vessels and GI movements. Brain, 3 major divisions. 1. Hindbrain, A. Medulla oblongata. It governs the respiratory and CVS. Regulate reflex activities, for example salivation, swallowing, vomiting, etc. Modulation of GI functions. Highway for all tracts of nerves that leaves and enters brain. Give rise to all cranial nerves except for smell, sight, and muscle of eyeball. BPONS. It relays information from medulla to higher cortical centers and cerebral cortex to cerebellum. Regulate respiration. C. Cerebellum. Involved in synergistic control of skeletal muscles. Play important role in coordination of voluntary muscular movements. Involved in maintenance of posture and equilibrium. 2. Midbrain. A. Cerebral peduncles. Involved in motor coordination and postural reflexes. B. Corpora quadrigemina. Relay center for hearing and visceral reflexes. C. Red nucleus. Regulate motor functioning. D. Substantia nigra. Regulate motor functioning. E. Reticular formation. It control or influence alertness, waking, sleeping, consciousness, and visceral reflexes. 3. Forebrain. A. Thalamus receives all sensory stimuli except olfactory, then relayed to cortical areas. Impulses are also received from cortex, hypothalamus, and corpus striatum and relayed to visceral and somatic stratum, involved in the integration of emotional behavior. B. Hypothalamus regulates body temperature, food intake and satiety, electrolytes and water, sleep-wake mechanisms, cardiovascular functions, carbohydrate and fat metabolism, secretion of hormones from pituitary organs. Important for integration of sympathetic and parasympathetic system. C. Cerebral cortex. Analyses sensory input, store information, memory development, analyses and control muscle coordination, hearing, vision, etc. D. Corpus striatum, responsible for modulation of motor movement and control of muscle tone. E. Rhinencephalon. It is concerned with reception and integration of olfactory impulses. 4. Basal ganglia. They receive inputs from the cerebral cortex and project output back to the motor and premotor cortices, responsible for integration and regulation of motor activity and postural reflexes. 5. Reticular activating system. It is alerting system of brain and maintain a state of alertness or arousal. 6. Limbic system. Control emotional behavior, feeling of anger, rage, pleasure, and fear. Regulate biological rhythms like sexual activity, feeding, and learning. Neurotransmission, neurotransmitters, NTs. Agents which are released from the synaptic vesicles upon the arrival of an action potential, for example NEH, etc. Neurohormones. Chemicals released by neurosecretory cells of hypothalamico-hypophyseal circuits into the blood, for example peptide hormones. Neuromodulators. Substances that can influence neuronal activity in a manner different from NTs. Neuromodulators originate from cellular or non-synaptic sites, for example adenosine and other purines, PGs, and their metabolites. Neuromediators, substances that participate in the elicitation of the postsynaptic response to a transmitter, for example involvement of CAMP, CGMP, IP3, DAG, etc., as second messengers at specific site of synaptic transmission. Neurohumoral transmission, transmission of message across synapse and neuroeffector junctions by the release of humoral substances, that is NTs. Impulse conduction, passage of an impulse along an axon. Impulse transmission, passage of an impulse across a synapse. Nerve action potential, electrical changes during excitation of nerve membrane. Events in neurohumoral transmission, release of NT on the arrival of an action potential. Combination of the NT with postsynaptic receptors. Initiation of postsynaptic potential, that is EPSP or IPSP. Removal, destruction of the NT through reuptake, metabolism, or both. Classification: 1. Conventional non-peptide transmitters, I. Quaternary choline esters, for example, OCT2. Amines, for example, noradrenaline, adrenaline, dopamine, 5-HT, and histamine. 3. Amino acids, for example, excitatory, glutamate, aspartate, and homocysteate inhibitory, gamma-aminobutyric acid, GABA, and glycine. 2. Neuropeptide transmitters, I. Peptide that act on brain and gut, for example, enkephalin, metenkephalin, substance P, gastrin, cholecystokinin, vasoactive intestinal peptide, VIP, neurotensin, insulin, and glucagon. I2. Pituitary peptides, for example, B-endorphin, amelanocyte-stimulating hormone, prolactin, luteinizing hormone, thyrotropin, growth hormone, vasopressin, and oxytocin. 3. Hypothalamic-releasing hormones, for example, thyrotropin-releasing hormone, luteinizing hormone, releasing hormone, and somatostatin. IV. Other peptides, for example, angiotensin II, bradykinin, carnosine, sleep peptides, and calcitonin. 3. Other mediators. I. Diffusible mediators, for example, nitric oxide, carbon monoxide, arachidonic acid metabolites, PG, thromboxane, and lipoxygenase. 2. Purines, for example, adenosine and purine nucleotides, ATP and ADP. 3. Cytokines norepinephrine have both excitatory and inhibitory action in CNS through alpha or beta adrenoceptors and B adrenoceptors respectively. IT deficiency in CNS may result in depression and its excess cause mania. Excess release in CNS results in arousal reaction, wakefulness, and alertness. Amphetamine and dopamine drug cause release of norepinephrine and produce arousal reaction. Reserpine depletes norepinephrine storage and produce depression. Sympathetic nerve originating from CNS regulate BP. Norepinephrine through stimulation of A2. Adrenoceptors in medulla causes hypotension. Tricyclic antidepressants, cocaine, amphetamine, clonidine, and methyldopa produce their effect through alteration of norepinephrine transmission in CNS dopamine. Dopaminergic neurons in CNS release dopamine. This neuron control both motor and behavior changes. Dopamine produce its effect through stimulation of D2 receptors. Dopamine deficiency result in Parkinson's disease. Its excess result in schizophrenia and Huntington's chorea in human. Its release form tubuloin fundibular dopaminergic neurons inhibit release of prolactin and increase secretion of GH from the anterior pituitary. It also cause nausea and vomition through stimulation of D2 receptors, CTZ. 5-HT, released by tryptaminergic neurons. It produces its effect through stimulation of 5-HT receptors. It causes hallucinatory behavior, antidepressant effect, and pain perception. Its secretion rises body temperature. Ache. Acts on nicotinic and muscarinic receptors. CNS effect is mainly due to muscarinic receptors. Enhanced cholinergic activity in association with excess dopamine activity cause Parkinson's disease and loss of cholinergic neurons in CNS results in dementia. Alzheimer's disease type in man. Amino acids, glutamate, aspartate, and homocysteate mediate excitatory transmission in synapses. Excitatory amino acids may have role in genesis of epilepsy. The inhibitory are GABA and glycine. GABA is the main inhibitory transmitter in brain. It acts through postsynaptic GABA and presynaptic GABAB receptors. Picrotoxin produces convulsion through blockade of GABA receptors, and diazepam causes depression through facilitation of GABA transmission. Glycine is the inhibitory transmitter in spinal cord. Strychnine produces convulsion by acting as glycine antagonist, and the tetanus acts by interfering with glycine release. Histamine. Histaminergic neurons in CNS acts through release of histamine by stimulating its receptors, H1 and H2. H1 and H2 receptors activation mediates excitatory and inhibitory actions respectively. Its role as neurotransmitter is not understood properly. Neurotransmitters.